Well, good afternoon, everybody. It's a pleasure for me to introduce today's speaker, Professor Agustin Ruiz. And Lhasa, I didn't see that one before. Is that your last?
You know, in Spain, we have two last names.
Okay.
That
And as you can see, he's an MD as well as a PhD. He was born and raised in the province of Sevilla, I think, and then went on and got a MD degree first of all in 1987 or something like that. And then after 5 or 6 years getting that degree, do you guys start straight out of gymnasium and go into that program?
exactly. We finish the gymnasium and we say with pre-university course and then move to the School of Medicine.
Yeah.
And when we finish, we can start a PhD.
Yeah, okay. And then he went on to get a PhD. Both of these were received at the University of Sevilla in molecular and cellular biology program. He's been recruited here to the bigs because of his international reputation in Alzheimer's disease research. And that's indicated by his incredible CV. This is like 1/100th of it. Just covering up bases here. Indications of the meaningfulness of his research is the fact that I went back and looked at Google Scholar citations, and he has about, I don't know, close to 10 papers that have been cited more than 2,000, 3,000, 4,000 times, and H-index, depending on where you look, of 52 or 76. So, and his work has spanned the gamut of research in Alzheimer's disease, with a big focus on, improving diagnosis and even treatment. What else do I want to say? I always am impressed with the mentoring part of a person's CV, and he's mentored 9 PhD students, all of them. It sounds like they've been excellent students that won top awards over the course. So I think he's going to bring that mentoring skill to us. And he is affiliated with biology of aging discipline. I'm very happy to have him as part of that. Oh, funding. Well, over the course of his years, he's gotten upwards of, I translated euros to dollars, $33 million in funding. And we expect that.
It's a lot more now.
Yeah.
The dollar is
20% more.
yeah, the dollars is really weak.
Yeah. And
I love that we improve in the future.
also in terms of papers, he's got about 300 publications to his name, and 97 or so of those are either first or senior authored, co-senior authored. That's a really impressive amount. So I think I'm going to stop there so that we can have as much time as possible hearing about his work. I won't read the title since it's there in front of you. But anyway, Agustin, so glad to have you here.
Thank you. Thank you very much, and thank you for the invitation. As Dr. Nelson has mentioned, I just landed in the States. I am learning about, I am in my learning curve about the way of thinking of the researchers, the way of doing research, the way of writing grants, and so forth, so on. So very glad to be here. I know very well also the reputation of this incredible institute, and I'm very pleased to be here. I'm going to introduce you a topic that is my very first time presenting these results, that is unpublished yet. We only present one communication in Alzheimer's Association International Conference 2025. So yeah, we hope to publish this soon. I hope you find this interesting. So yeah, I'm going to speak a little bit about the proteogenomic analysis, and I would say the proteomics, I would say omic landscape in the cerebrospinal fluid of humans, okay? So, yes, as mentioned, I have my PhD on molecular biology, molecular and cellular biology, but I'm mostly focused on genomics and all my entire career. My dream was, to convert the genetic information and its connection with the human diseases in something instrumental for our patients in the future. So we started researching providing linkage to new genes associated to diseases, and now we are trying to understand that genes. So that is my mission mostly. And of course, I have a perspective of a geneticist, not only for Alzheimer's disease, but for any human disease, of course. What is interesting to me as a geneticist is that most of the cases of Alzheimer's disease are considered multifactorial or complex. That means that there is an interaction between genes and environmental factors, now called exposome. This is the new fancy word we are using for the non-genetic factors as a whole. Also interesting and connected with your institute is purely age-related. So that means that the most important risk factor for Alzheimer's disease is aging. Okay. I like very much this disease, to study this disease, the disease I don't like very much, I have to say, is because it's also a very, very genetic related. The heritability of the disease is very, very high. That means that the proportion of factors associated with Alzheimer's disease, genetic factors are very, very high, more than 60% of the variance of the associated with Alzheimer's disease is genetics. So that means it's a good model for looking for the connections between the genes and complex disorders. There are only a few cases of Mendelian mutations provoking the phenotype, but it's only affecting to 1 to 5% of the cases, depending on the series. So as I mentioned, most of the cases are a combination of exposome and genome of the individuals in a model that we don't understand well yet. Of course, from the medical perspective, this is a very important burden for our society. And you can see here, general figures, 57 million people afflicted right now. And this is going to double every 20 years. And we have no solution for this disease. So we need to look for, new ways for combating the disease. Okay. Diagnosis, the problem with Alzheimer's disease has been, defined by physicians like me. That means that we are, just making the observation of the phenotypes in the microscope. And we saw 2 specific deposits, amyloid, extraneuronally in the matrix, with plaques of amyloid, and we have intracellularly in the neurons, we have tangles of phosphorylated tau. And that is the definition of the disease. When you have these deposits in your brain, you are endorsed with the diagnosis of Alzheimer's disease. And we don't know why these deposits are occurring in parallel with the disease. And even we don't know if the deposits are causative at this moment. But it's our definition. It's what we have. It's a pure observation in the microscope of the brains afflicted of Alzheimer's disease. We have a progressive neurodegenerative disorder. So we are losing neurons because of this deposit. It's supposed to be because of this. And this is provoking, the loss of independence of the people. So the social burden is massive because people at the end of the process cannot eat alone, they cannot dress properly, cannot, take care of themselves. So this is a massive burden for the family and for the society. And we have no treatments. This is just to reinforce, because if you want to focus on the genomics of complex disorders, you have to take into account the measurements of heritability. This is an example that actually the genetics of the complex disorder is interesting enough for focusing on it. It's dependent on the variance of the phenotype explained by the genes. And you can see here that one of the most influential genomes in relation with the disease is associated with Alzheimer's disease. But you can see here also this big purple bar that is also increasing here. This is aging and sex. But what we understand is that portion of the genome is also gene sex and gene age interaction. So it's probable that this part of the bar and this part of the bar are because of the interaction of the factors among them. So the genes probably are also connected with the aging process, of course, and with the environment, toxicities or any other loneliness, for example, as a risk factor for dementia is well established. So there is a connection that we don't understand well between all of these partitionings, I would say, of the variants of the phenotypes. But genetics is strong in Alzheimer's disease, and that is a good reason for focusing in the decoding of the genome landscape associated with the disorder. That was one of my turning points in my career was deciding to go to Barcelona. And I was, as I mentioned, I am very interested in the genomics of complex diseases. And in Barcelona, they had a growing biobank of cases. This is probably the single site bank or biobank for DNA, the largest in the world, okay? And that was a good reason for joining them and start working with this massive amount of samples recruited in a single site in Barcelona, Spain. So this is, I need to update this. We are approaching right now 28,000 people with genomic information available in our institution in Spain. And this is the real legacy, and we are exploiting this information the most we can, trying to deconvolute the genetic intricacies associated with the Alzheimer's disease.
So what is the population that this is drawn from?
This is our reference population is Barcelona. We work for the public health system there, and we are, the health system is different in Spain, it's public, so and universal. That means that we offer our services to anyone. So we are probably the largest memory clinic in Spain. We are attending about 8,000 people a year, 2,000 new patients a year, and we are collecting samples from all of them. That's the reason why, these numbers are growing and growing all the time. Of course, we are not only taking the samples, we are deconvoluting using array analysis, sequencing, deep phenotyping, molecular phenotyping, and we try to put together everything and make interpretations and looking for new discoveries associated with Alzheimer's disease. So, this is, I would say, the temporal line of our program. When I arrived there, I made the decision 2011. They offered to me, something. They have the biobank, but they have no wet lab, no dry lab. They have no internal research. And they offered to me, okay, Agustin, you wanted to come and build up from scratch a new laboratory? I say yes. And I say, yeah, because the amount of samples available, okay, I need to decode the samples and see what is behind the genetics of Alzheimer's disease in our population. So we built up the laboratory. We also convinced a group of people, you have seen, that for doing the same in the whole country. So we joined all the 12 different groups, prepare a single protocol, and start collecting samples, not only in Barcelona, but in the entire country. This is the Dementia Genetic Spanish Consortium. That is a consequence of people who wanted to do the same we did in our institutions. And yes, we start decoding the DNA samples and also connecting our operation with other studies, especially in Europe, but also in the States, and start running metageos for identifying genes associated with the disease. This is not because of our work, it's a cooperation, a global cooperation. We are part of the European Alzheimer's and Dementia Biobank, which is the largest operation identifying genes for Alzheimer's disease. But because of the statistical power we supply to the consortium, we start getting the leadership of many of these papers as well. So we are part of the steering committee of the European Alzheimer's Disease Biobank. We are very last or very first authors of that paper, of course. So yeah, and this is because of the statistical power of the Spanish data set. It's very, very strong. Okay. I don't want to talk about this. This is to mention that we are identifying the genes associated with dementia and Alzheimer's disease. Okay. And this is a spoiler, but I think you are recording for YouTube, but this is not published yet, but this is the very last landscape is going to be published probably in upcoming months. It's under review, mostly accepted in Nature Genetics. And these are the 91 genes associated to Alzheimer's disease available. Available means that, okay, we have the hits. We don't know anything about that hits. We don't know how they work, how they can be connected with the pathology of the disease. We have no idea. So long way ahead, and we have to back to the bench and start delineating experiment to understand the connection of this loci with Alzheimer's disease and related dementia. And that is the reason why I'm here now. I think we have more than enough genes that we don't understand, and I wanted to translate the information coming from the genome in something instrumental for the patients. And for doing that, we need to back to laboratories, start modeling these genes, and start understanding why these genes are associated with Alzheimer's disease. And this is my work here. Okay, okay, let's go to the meat of the matter. As I mentioned, we are part of the European Alzheimer and Dementia Biobank. This is already published in 2022. This is the very last release of the European Alzheimer's Disease Biobank. And you can see here in the Manhattan plot a massive landscape of hits. So we have a lot of candidates to connect pathobiologically with Alzheimer's disease. But if you remember at the beginning of my dissertation, we have a definition of Alzheimer's disease based on the amyloid burden extraneuronally and the tau burden intraneuronally in the brain as a definition of Alzheimer's disease. And you can see what is happening. Most of the genes that we connected with dementia in clinical samples are not associated with Alzheimer's disease when we look for the endophenotype defining the disease. Why? Probably we have not enough statistical power. This is a very different study. Here we have 750,000 individuals in the study, and we have here only barely 13,000. But you know, the genetics of I would say quantitative loci or quantitative proteins is much more powerful than conventional case control. So we need to see much more here and have to be very well aligned with this. We have barely one hit in common, ApoE, and we have another in common, is CR1 for A-beta-42 levels in the CSF, and another being one for phospho-tau levels in the CSF. Where are the other genes? We don't know. One explanation is that the definition of the disease is based on epiphenomena and are not causative. Another explanation is probably the lack of power of these experiments. We are increasing power. We plan to release a new version of this association to the main endophenotype of the disease, and we are start seeing more hits, but not with the density of hits we find out in the conventional case control. So, well, but what is most important or most interesting from my point of view is this important hit we have here for phospho-tau. This is a gene with a very, very strong effect in the level of phospho-tau in the cerebrospinal fluid of the patients, but it's not associated with Alzheimer's disease. And these have a very deep implication for the understanding of the disease. How is possible, if the phospho-tau levels are toxic in the brain, how is possible that the main gene controlling the expression levels is not associated with the disease. We don't know why. But it's again pointing to the notion that actually these protein levels are mere epiphenomena associated with the disease and not causative. That is something we need to understand better in the future. But that's the reason why, we change the surprise to something like we are going to start being more astute about these findings and look for what is behind this mismatch between the clinical definitions.
My question is just to make sure I understand this, that earlier, the hits that are in this most recent GWAS, that those are not showing up in the genes like with the two biomarkers that we have defined Alzheimer's by. And so the question is, why not? Is our definition incorrect? Or where are the genes and do we not have enough power? Is that what your premise is?
Well, I have to say that I don't know. This is the mismatch need to be investigated. But one possibility is, as I mentioned, lack of power, because here, as mentioned, 750,000 people in this experiment, and here you have only 14,000. But again, measuring the protein levels and combining that information with the genome, we know we have much more power for detecting genes associated with the quantitative level of protein than with the conventional case control analysis. If you make appropriate power calculation, this is equivalent to about 200,000 people in a conventional case control. So we expect to detect a good proportion of these genes associated with amyloid beta 42 levels and phosphatase, and we are not identifying such a connection. So, this is an open question. We have no answer for this, but we suspect that probably what we are seeing in the microscope is a very, very long-term consequence of a process. And the etiology of the disease depicted in this landscape is connected with processes that are occurring in advance of the amyloid and phosphatase accumulation in the brain. That is our suspect, but we need to demonstrate. So, yeah, we are going to try to deconvolute the connection between genes and the disease. We are looking for new sources of information. And what is closer to the brain that we can take regularly in our patients is the cerebrospinal fluid. So we are, running massive experiments trying to deconvolute the omics of this cerebrospinal fluid and trying to connect the patterns at the different omic levels with the biomarkers of the disease, with the progression of the disease, with all the definitions that have been provided by neurologists and neuropathologists. So that is our, and how it's produced, what is interesting, this is, for example, the protein landscape in the CSF is in some way mimicking what is happening in the blood, because the choroid plexus is producing the CSF, the cerebrospinal fluid, filtering in some ways, filtering the bloodstream arriving to the choroid plexus. So 80% of the proteins that we have in the CSF are coming from the blood. But 20% are coming from the interaction between the CSF and the brain parenchyma. So that means that probably the different cells are also putting a signature of their own protein into the CSF. And that's what we are researching, trying to separate the gold from the dross in the CSF, trying to, I would say, looking for the connection between all the omics landscape we can capture from the CSF and trying to connect that landscapes with the AD genetic landscape I showed you earlier, okay? So for doing this, we need to understand well the way the CSF is produced by the choroid plexus, but also the way the CSF is also reabsorbed by the arachnoid granulation, for example, or by the nasal claviform trains. By the way, this is the reason why it's very effective, the nasal pathway to reach the central nervous system. It's because of this clear connection between the uniform plate and the brain. Okay, so we need to understand this physiology and the physiopathology that we can identify in the I would say in the biofluid CSF. As I mentioned, is the closest bioresource we can get from our patients regularly. Okay, here is just to see you the structure is the correct plexus. This is the main structure in the brain, is in the ventricles, producing the fluid that is circulating in the central nervous system. Okay? So, yeah, that was a technical decision. We get money from the different agencies and decide for starting the convolution of the CSF of our patients. And this is one of the first experiments we ran in Spain. This pure brute force, take the CSF from our patients, define clinically them, define from the biomarker point of view, from the progression rate point of view, blah, blah, and check on start applying omics to the CSF to see if we can detect any pattern associated with the endophenotype, with the disease progression, etc., and etc. I'm mentioning this because, this is the reason why we are very successful with the papers, and we publish many times in Nature Something, is because of the cost of the experiments. This is a $2 million experiment, okay? And this is, I think, $750,000 experiment, okay? So that's the reason why, when the editors take these kind of big experiments, they say, oh, wow, they have to come here, etc. That's because of the ultimate technologies, applying multi-omics to humans and etc. is very, very, I would say, brute force and fancy experimental approaches that are very, very nice for the editors. But the thing is, we are producing these brute force experiments, but we don't understand a word what it means. Probably next generation of bioinformaticians will help us to understand the pattern we are obtaining. Okay, and that is our role here. For example, in our dry lab, we are concentrating this kind of experiments and look for hidden patterns and appropriate interpretation of our results. So I will show you a combination, integrative analysis of these three experiments and what we take from our patients. Okay. And again, of course, our collection is growing. This was the first wave. We started running in 2016, our CSF cohort, but now we are approaching more than 3,000 CSF specimens of different individuals ready to go for further experimental approaches and increasing the brute force more and more in the future, increasing statistical power and increasing resolution at the omics landscape in the patients. Okay, so I will be, playing around the information of these three experiments. This is Lipometrics, is a company that convoluting the lipidome of the CSF. This is a European company. Probably most of you know Somalogy. This is a massive proteomic platform. In our case, we ran the experiment with the 7,000 protein forms in the CSF. And of course, we combine information of the omic landscape with the genetic information of all patients. So all the patients have the very same, I would say, experiments and the very same information behind, from the clinical, biomarker, and molecular point of view. As I mentioned, because the people know we have that kind of things, we are invited many, many times to be part of massive operation trying to convolute the information of the omics landscape in Alzheimer's disease. So we are cooperating with a number of different groups around the world. We have a very, very interesting cooperation with Washington University and a group of Carlos Cruchaga there, because they are very interested in the proteogenomic analysis of the CSF. So the idea is to generate a map or a landscape of all the modulators of all the proteins expressed in the CSF. And we have a massive inventory of genetic regulators of the protein expression in the CSF of humans, and trying to identify in that way the connections between the alleles associated with the disease and the differences in expression levels and its connection with the phenotype. That is the philosophy of the proteogenomic analysis. Now we are refreshing also this study. Probably next year we will update the landscape, adding, I think, 1,300 new loci controlling the protein levels of the disease. And you can see here, there are two different kind of regulators of the protein levels. One is, this line is describing the cis regulators. So the markers, the genetic markers associated with the protein levels surrounding the loci encoding the proteins itself. So this is cis acting elements. And you have the other points that are outside the line are trans elements. So there are other points in the genome not close to the coding sequence of the protein that are regulating in trans regulators of the protein expression. Okay, so this information is growing and growing, and we are using that information trying to run Mendelian randomization studies looking for causality. So trying to connect the alleles with like specific protein levels and to check if these changes in the protein level are the cause of the connection with the disease using Mendelian randomization approaches. And of course, the people is trying to predict with the protein landscape and other omic landscape to run differential diagnostics experiment and other kind of experiments. Looking for more precise diagnosis of the disease. And this is the very last consortium that have been, we are part of that as well, trying to collapse. We are switching there from $2 million experiments to $30 million experiment. This is the case of the Global Neuroproteomics Consortium. They are concentrating in a single consortium the protein profiles of more than 40,000 people. Okay, so the idea, again, is a new exercise of brute force, but again, long way ahead to understand the patterns we are now, providing to the scientific community. A long way ahead to understand the connection of these proteomic landscapes with the diseases. So we are just producing and producing more and more data. Sometimes the science is not as fancy as I show you in the Nature Science papers. And when we start working in our own data sets, we start detecting, we have the benefit of having very good friends, and we have sometimes overlapping between the proteomic experiment run by one or others. And we can try to reproduce in the very same sample, even the same platform is producing this kind of bimodal distributions. That means that a big proportion of the protein measured by SomaScan in this case cannot be reproducing in a secondary experiment with the very same sample and the very same platform. That means that a big proportion of the measurements are not valid. Simply. Believe me, it was very difficult to publish these results, as you can imagine, because only with this graph, we are destroying a big proportion of the SomaScan papers that have been published out connecting proteome landscapes with Alzheimer's disease. But we try also to reproduce in independent platform. And again, the bimodal distribution, comparing SomaScan with Olin was massive. That means that a big proportion of the results measuring the protein with one method or another are not matching at all. So that means that we need to separate the gold from the dross also in the signals coming from the proteomic landscapes of these platforms. And we are trying to do that. And of course, trying to look for the bias associated with the I would say, the landscape we are taking from our patients. So we start realizing that we have very cumbersome, for example, volcano plots. When we try to compare the proteome or to identify protein associated with the amyloid burden in the CSF, this kind of escape with volcano plot, we don't like very much. And look at for the inflation, the statistical inflation, 62.3% higher number of positive than we expect by a chance. This is very cumbersome for people working in statistics. So we need to understand this kind of bias, and we need to implement this information to have a better idea what is actually important in the profiles we are detecting. Okay, it's just what we are doing. And for example, we say, okay, we have this kind of very strange statistical behavior of our landscapes. Why not start comparing, what is happening for the very same individuals in different omics? And you are seeing here, this is I would say every point or every line is representing a patient. Here you have the samples separated or ordered by the mean expression of the lipids, mean expression of all the lipids we detected, okay? And also sorted by the lipids, the C score. And we did the same for the protein, the very same individuals. And you can see here a common pattern. So that means that there's something in the CSF, something that is differentiating the burden of most of the analytes we are measuring irrespective of the platform, lipids or proteins, and are connected with anything is a main component of the variants that we don't understand. And it's in common in different omics. You can see here, probably reaching this kind of p-values is not easy, There is something biological or that there is a specific artifact, could be a statistical artifact or whatever, that is affecting to the whole landscape of different omics. So let's try to dissect this. And this has been our work during the very last 2 years, trying to understand this. So we started analyzing the principal component analysis and start comparing the principal component analysis of the different omics, and also comparing that with the mean value of the, I would say, the brightness of the samples in any different omic. And we realized that most of the variants were associated to two principal components, irrespective of the platform we are using, is irrespective of the analytes we are wanted to detect. And we have connection of this principal component 1 and principal component 2 of the two different platforms, one measuring lipids and the other measuring proteins, and you have a common variance among them. Why? Why is happening this? At first glance, we say, this is probably an error. This is something something is wrong. This is a kind of bias we have in our study, and this is not biological, especially because of this strong correlation and the p-values associated. So we wanted to go in more depth this behavior of the different omics platform in our hands, and start saying, okay, let's analyze the factors, or let's deconvolute the factor associated with these principal components of the variance of the different omics experiments. And you see here what we find out. The first we find out is that the first principal component, the most important one, was unrelated with the disease, but surprisingly was strongly related with the amyloid and phospho-tau levels. How is that possible? How is it possible that one component of the variance strongly associated with phospho-tau and amyloid is not associated again with the disease? It's something to be thinking about, okay? And we found out the second component was barely associated with the dementia continuum. If you compare the mean values for the controls from the people with an intermediate phenotype we call mild cognitive impairment and dementia, you have association, at least of the principal component 2 with the disease. That means that probably behind principal component 2, there is something that is meaningful from a pathobiological point of view. But what is behind the principal component 1, despite it's associated with CSF, phospho-tau levels, and amyloid, is not associated at all with the disease. So let's try to continue the convoluting the signal we have behind principal component 1 and 2, and we did. We're back to genetics. Genetics is interesting because it's very powerful because the germline variants are static. So we have no problem with variability along the time and that kind of thing. You have your DNA profile. And we also, okay, if there is something behind principal component 1 which is biologically meaningful, we need to look for any kind of correlate at the genome level. If not, probably it's merely an artifact of the experiment. But what was fascinating is that we detected genes associated with the principal component. So that means that there is something biological explaining this component of the variance of the proteome and lipidome landscape in our CSF samples. And you can see here exactly the very same signal, either in the protein landscape and the lipidome landscape. The very same signal as the GMNC locus. Okay, we detected other signals in other principal components that also make sense. For example, this signal in FADS1 associated with the lipidomics profile is a fatty acid desaturase 1 that is very well established biomarker of the lipid levels. So we are detecting biology behind this. This is at the end is the message I wanted to bring you. So okay, we start analyzing the GMC gene, and we detected that it was curiously associated with the volume of the brain ventricles. And for us, make a lot of sense. Depending on the size of the ventricles, the levels of the protein can be different because are more or less diluted. If you're producing the same amount of proteins, but you have to dilute in the largest volume, most probably, it's a good explanation of the reason why we have different levels of multiple proteins in different individuals. And it's because of the differences in their own anatomy and probably in the production of the CSF itself. Because we realize that GMC is promoting multiciliated cell differentiation, which is the key cell connected with the production of CSF in the brain. So we identify all their genes that are in literature associated with the ventricle volume of the brain and test the hypothesis as well. And we demonstrate that all the genes associated with the volume of the ventricle were also associated with the principal component 1 and 2 we detected in our profiles. So that is in a way proven the idea that actually the level of multiple analytes in the brain are merely depending on anatomical and physiological differences among individuals. Okay. So this is the very simple explanation behind our findings. You have larger ventricles, you have diluted everything. And if you remember, the top hit in phospho-tau, unrelated with Alzheimer's disease, we published years earlier, is GMNC. So that means that we declare a gene controlling phospho-tau associated with Alzheimer's disease that actually is associated with the levels of many, many proteins in the CSF. And it's not a regulator of phospho-tau at all. It's a regulator of the amount of proteins you have in the CSF. So now we understand why we have a gene associated with phospho-tau unrelated with Alzheimer's disease. Okay. So let's dig a little bit more on the proteomics profiles and on the top hits in the different principal components. So we try to identify up and down regulated genes associated with these principal components, okay? So the idea was to run some kind of enrichment analysis comparing the top co-regulated with principal component 1 and 2 and top down regulated with principal component 1 and 2 and see what kind of functions come up doing this kind of analysis. Look at this massive signature of principal component 1. More than 1,400 proteins are connected with GMC and other genes associated with the ventricle volume. And only 10% of this amount is non-regulated. It's going in the opposite direction. How that is possible? We mentioned that there are a dilution of the proteins because of the enlargement of the ventricles. How is possible there are also proteins in the opposite direction? Let's dissect the functions of this. We conclude based on our analysis of the top up and down regulated proteins that the PC1 upregulated component, which is mostly diluting the burden of proteins of more than 1,400 proteins, are associated with protein highly expressed in the central nervous system. So this component, this physiological or anatomical characteristic, we have not proved to decide is anatomical or physiological or both, we don't know, is affecting to a large amount of proteins that are mostly expressed in the central nervous system. In the other way around, the proteins that are downregulated are coming from the blood, because it seems that the GMC is connecting in a way also the active transport of proteins, so that people have a big clearance of the protein, but the correct plexus is also acting or activating the transportation of the protein into the brain as well. So most of the downregulated by PCF1 proteins are associated with circulating proteins in blood. The other way around, most of the proteins that are diluted in the brain are connected with the protein produced by cells in the central nervous system. And you can see that, of course, the genes associated with the ventricle volume, that are here in blue and here in orange, are operating in the very same way with the principal component 1, but in opposite direction. A little bit complicated, but at the end, with the enrichment analysis, we detect that the two components in principal component 1 is associated with the brain-derived proteins, and the other is associated with blood-derived proteins. And that happened with principal component, yeah.
So a little bit confused. So if you think, speculate that's affected by the volume of the ventricle increasing, if you use it as a confounding factor to correct it, will you be able to detect A-beta-42 and phospho-tau?
We will continue.
Okay.
But you will see. You will see what happened and the implications. Principal component 2. Again, we ran enrichment analysis and detect that most of the protein associated with principal component 2 are coming from the blood as well, and interestingly, associated with a disruption of the blood-brain barrier. Remember that principal component 2 was associated with the disease continuum, and we know this is a well-established factor of evolving factor in the disease is the blood-brain barrier disruption. So we think we have found out the proteome signature of the blood-brain barrier alteration in the profiles of our individuals. And this principal component 2, as you can see here, are associated with the risk factor for the blood-brain barrier dysfunction, okay? So how to use this information? That is the practical stuff related to biomarkers. So, yeah, we need to correct our proteome and lipidomes and NEOM landscape. I would say NEOM landscape in the CSF, but principal component 1 and 2. That is very necessary. In order to reduce the lambda, the inflation of the R statistics. Look here that for the phospho-tau, we have an inflation of infinite in our study. And adjusting by principal component 1, we reduce from infinite to only just 14.9. Okay, so we are reducing the statistical noise, reducing the artifact, and probably the genes or the protein associated with the disease are much more reliable in this experiment than in this experiment. You have less noise. And the very same for adjusting by principal component 1, the A-beta 42 levels as well. You can see this cumbersome, I would say, asymmetric volcano. We don't like very much because of this inflation. Just disappear when you correct by principal component 1 the proteins associated to amyloid. So probably these results are much more valid than this one for making appropriate interpretation about the proteome associated with amyloid in our CSF, okay? So yeah, we need to prove that. So we look for the way of identifying within the proteome, reference protein, correct biomarker levels, correct NEOmic experiment. And you can see here in our hands, when we start adjusting, for example, the biomarkers by these new components, we increase the prediction capability of the biomarkers, and we have more precision identifying people with amyloid and tau positivity in the brain. You can see here how using these potential candidates for correcting the experiments, we are improving the area under the curve for disease progression, is improving, and also is reducing the statistical noise, the lambda inflation in our experiment. So yeah, these have important implications because if you consider the same cutoff for everyone, but you have different levels of protein burden because of physiological differences or anatomical differences in individuals, this very same cutoff is producing misinterpretation of the positivity of the biomarkers. And we detected that 67% of the people with that mismatch in amyloid and tau positivity was because they have very high concentration of the proteins that we are using up now as an adjusting protein for our measurements. And the other way around, the mismatch of amyloid positivity without tau positivity was connected with low concentration of these proteins in the CSF. So we need to correct. If you wanted to use a single cutoff for saying which CSF is positive or negative for biomarkers, you need to correct by the physiological amount of proteins of the individuals. And this is, my very last slide. This is a paper we are collaborating, but it's, I'm not part of this investigation. And for me, it's very funny because you can imagine the implication of this research for all the proteome landscape of all the protein inventories we have literature. You have not corrected by this kind of general factors. You are producing a vast amount of false positives. False positive protein associated with the disease and associated with amyloid and tau burden. And you can see here the differences of the profiles. When we get in this case, the landscape associated with Alzheimer's disease, autosomal dominant Alzheimer's disease, a big proportion of these proteins were false positive because they were mostly associated with principal component 1 and 2, and not associated with Alzheimer's disease, autosomal dominant Alzheimer's disease. So this is something that my feeling is that this presentation is finishing. It's finished mostly. We will have a lot of difficulties for publishing this because we need to reanalyze a lot of data because of this information. So conclusions, you see that Alzheimer's disease research is evolving very rapidly. Now we have 90 genes associated with the disease. We don't understand most of them, so a long way ahead to convert that in something instrumental for patients, but at least we have the genes. So we can start working with that. The need of the discovery and the convolution of the endophenotypes is very important to understand the disease pathobiology, of course. We detected in our study 2 biological axes, both are biological, so they are not a statistical artifact. One of them is related to the production itself of the CSF, and the other is connected to blood-brain barrier integrity. And the patterns we have detected are consistent in 5 independent experiments. And now we are preparing our paper, also contrasting the information in our dataset with others. And we are detecting the very same in different cohorts. So we have consistent, I would say, consistent effect of principal component 1 and 2 in different, I would say, experiments. We need to adjust for these latent factors. Otherwise, we will go for misinterpretation. And of course, we need more integrative approaches for the convoluting heterogeneity. And of course, for brute force, we need a lot of money. So we need to burn more and more money the convoluting our samples in the future. So, and that is my very last slide. This is the people doing the work, and me. I just presenting this work. And these are my, my very last PhD students in Spain. Pablo Garcia Gonzalez is in charge of this investigation you have seen. And Raquel Puerta, she is in charge in the proteogenomic analysis of our population as well. And of course, the sponsors supporting me in Europe, and now I started supporting me in the States. Thank you very much.
So I'm curious about your CSF turnover. How do you measure it, really? I mean,
We cannot measure.
it's pretty small.
Yeah, of course. This is a next tax.
This is big. I mean,
Exactly.
not on this many people,
Yeah.
but you could. We can do it here if you want.
Yeah.
If you want to do it, we can
So,
put a tracer and then
yes, when I was creating the first draft of this paper, and also preparing the slides for the conference and we're showing you today, I was thinking exactly on that. We need to run a GWAS on the CSF production and clearance.
Yeah.
In our patients, but that is expensive.
you can use deuterated water.
Yeah, but the point is that we need to offer that technique to our patients and see if they accept.
Just drink the water, and then you do a series of CSF, a series of spinal taps, or you put a catheter and then you draw.
To reach 3,000 people or whatever.
It's expensive.
That's my point.
Yeah,
I was thinking on MRI-based proxies.
that's what I would.
So as soon as we inaugurate the 7 Tesla MRI, I am thinking in a proxy for large scale. But again, I agree with you. We need to have a proof of principle. Genome-wide association study, I would say 200 people with that experiment you have. And see if we can detect the GMC, for example.
Or you could do the opposite, so you could take the extremes of your
Yeah.
curves after you've done your experiments with GWAS and all this, and all the multiomics. And then you measure on the subset of subjects.
Yeah.
To see if that's real.
Yes, I totally agree. I think that we need to look for the way to research them for a direct evidence of CSF production. Totally agree, yeah.
Couple questions. So the first one is like, how much conservation is there between the hits you observed in like the European population compared to other populations?
Well, we have a lot of inequality on that, to say. We have more than 100 studies in, I would say, Northern European populations. I'm working with the Southern European population, we have less information there, so it's very skewed to the north. We have information because we are start, as I mentioned in my slide, we start collecting related population to the Spanish population, Chilean, Argentinian, for example, most of them are mixtures between Native Americans, Black populations, and white populations. We are detecting similarities. It's not exactly the same. For example, we have the polygenetic score combining all the hits, all the SNPs we detected associated with the disease are working well in different populations. This is another Nature something we published 2 months ago, looking for the translatability on the PRS among different ancestries, and it's barely working, but with some differences. For example, the scoring of the Chilean and Argentinian populations in our hand, the general scoring of the population is lower than the scoring we detected in Spain and other European countries. So, yeah, we are proposing that we need to adapt. We need to generate a tailored polygenetic score for depending on the ancestry background. We're working on that. But many of them are reproduced in other ancestors.
And then my second one is that how hard is it to be able to score out some of like the confounding factors, such like environmental factors that may be promoting these mutations? And
yeah. Well, another nature I don't believe is this one. You know, this is my very good friend Cornelia Van Duin in Ukebaba. For me, it's near impossible to think that the exposome, this is the green component in the bar, is as small as this in Alzheimer's disease. I don't believe that.
What's PRS?
PRS polygenetic score.
Oh, fine.
Is the genetic component. What happened with this is, you know, this is based on Ukebaba, it's based on 500,000 people. So you tend to believe that this partitioning is correct because of power of the study, you know. But actually, you know, you are measuring here half of the variance associated with the phenotype is because genetics. But when you try to go directly to the PRS composed by the 91 variants we already have, the variance explained is less than 2%. So how is that mismatch?
Also, I mean, if you look breast cancer, this whole genetics is
Yeah.
not, it is not, it's random a lot of
Yeah,
it.
but again, why is happening this?
Yeah.
Again, you know, I showed you this is to spur discussion on this.
It's actually big, breast cancer.
So the point is that because of the fixed, no, but the problem is here, you know, we have much more power to detect genetic effects, even without the gene environmental. By detecting the exposome with the currently available human experiments, it's nearly impossible. Why? Because maybe the exposure are affecting the people 30 years in advance of the symptoms, and we have no records. So we are capturing only the component of the gene environmental component, but not environmental. That is my interpretation. So we need to look for the counterparts.
Right. But it's also really skewed. I mean, you look at age and sex for breast cancer. I mean,
Yeah.
sex is a massive effect because it's 100 to 1 in women.
Yeah.
And age is another massive effect, and you can't see, I mean, if you would think it's all genetics.
Yeah.
From this, I tell you, Nature Medicine sometimes publishes stuff, but.
Well, we have a joke around, any nature something, you know, you have one paper in Nature Medicine and 1,000 papers saying it fails, you know.
Yeah.
But that is why you
Yeah.
get 1,000 citations.
Yeah, that's the way to do it.
So that's very true. So, yeah, that is, I have showed this to spur discussion. Probably, you know, we are modeling very badly the exposome. We're modeling a little bit better the genome. But at the end, with our genetic model, we are also explaining 2% of the 50% of the supposed variance explained by genes. So where is the missing variability in genetics? Where is? Probably in gene environmental equations that we don't understand. That's my modest opinion on this.
Could you go to that slide that showed the very limited number of loci that affect the two major indicators of the back, where you could, on the right,
You want this one?
no, farther at the beginning.
Okay.
It's the one.
This one.
This one.
So if you just randomly selected, what is it, 13,000 individuals from 64,000, how many of these would lose significance?
In this case, probably none,
None?
probably none, because of, you know, this whether you can see here, because in the next paper, we were checking, you know, the consistency of the signal when we are using just cases and control is the first dot. Excuse me, no, this is the whole meta-GWAS, including proxy AD. This is a UK biobank proxy AD is related to separating the people with and without familial antecedents of dementia, okay? And we are using that like a proxy phenotype, and it's working well, it's mimicking. So removing biobanks is the third dot, removing proxy is the second dot, and the first one is the whole stuff, okay? So you can see here how consistent are most of the studies. You have the three red dots in one of them, where you are detecting, you know, inconsistencies. Probably we need more power, more brute force, and probably we need to have better definition of the disease, because the major criticism we have in this kind of studies, and we are putting into the pot dementia. We are not putting into the pot Alzheimer's disease because of the low resolution of the diagnosis of our patients. But again, geneticists love have million people in the study. They don't care very much about the phenotyping is also important as well. But at the end of the day, you know, even with this sensitivity analysis, you can see how consistent is the signal. So removing a fraction of them probably is not affecting too much.
Another question. So like one of the hits you mentioned for like the proteomics data, the GMNC you mentioned is related to ventricle size, right? So I would imagine those would pop up when you have more severe atrophy. So how sensitive would it know? Even with like
It's not, this component is not associated with the disease. This component is associated with the anatomophysiological characteristic of the people, but not associated with the disease at all. Remember, the findings are, for me, are amazing. You can see the behavior when we compare controls, MCI, and dementia, but not significant for principal component. And for disease progression, nothing. So this is completely unrelated with the disease.
It does, just means that, the size of ventricle change is a dominant factor here.
Exactly. For the analytes we are measuring in the CSF, yes. And unrelated to the disease, yes. So we need to correct all of our studies. Any omic, you can find out, need to be corrected by the GMC unrelated, I would say physiological or anatomical components.
So how much would a protein have to change to be considered like a useful biomarker?
Okay,
For
that is depending on the way you are discriminating the people. Here you can see we are establishing comparing people with or without, I would say, clinical and biological definitions of the disease. We compare the level of the candidate biomarkers and look for those biomarkers separating better both categories. But the problem is that if we have this kind of component, these are tertiles of the principal component one. You see people who are controlled, you are seeing that for any reason they are escaping from the threshold for amyloid. And the other way around, they are entering in the threshold of phosphodiesterase. That's the reason why you have A negative, T positive. This mismatch is because of the differences in the amount of the protein naturally occurring in the people. And it's affecting, of course, to the interpretation of the results. We never understand well the reason why that was happening in our population. And now we understand a little bit better.
Well, thank you very much. Very sobering.
Let's continue.